Biology part 49. Regulation of plasma protein. Blood plasma contains about 70 gram dm cubic of proteins in colloidal solution. In addition to blood clotting proteins, fibronectin, these proteins include albumin and many different globulin proteins, covering a wide range of functions. Collectively, the plasma proteins act as pH buffers, and as discussed in this tutorial, they help to regulate the distribution of liquid between the blood and tissue fluid. Almost all plasma proteins are manufactured and later broken down by the liver. Number 4, regulation of lipids. Lipids are needed as building material for cell membrane and for making some hormones. The liver produces fatty acids and cholesterol interconverts. Existing lipid substances absorbed from the diet. Surplus lipids are stored as fat. Number 5, storage of vitamins and inorganic elements. Liver cells take up and store vitamin B12 and the fat-soluble vitamins A, D, E, and K. These substances are released whenever required. Iron is the main inorganic element stored, but the copper, zinc, trace elements including the cobalt and molybdenum are all stored in significant quantities by the liver cells. Production of heat. The liver is a major heat source for the body. Heat produced by all the chemical reactions going on to the liver, which is distributed to the rest of the body in the bloodstream. In mammals and birds, this heat helps to maintain a constant body temperature, thereby providing a near optimum conditions for the enzyme activity. Storage of blood. The liver acts as a blood reservoir, storing up to 1 liter of blood. The amount of blood retained depends on the diameter of the various blood vessels leading up to the form of leading to and from the liver, and this in turn is controlled by the nervous system and by hormones. During exercise, the liver provides extra blood, increase oxygen transport to the muscles. Excretory functions. Now we talk about the excretory functions of liver. Excretion is removal from the body of the waste productions of the metabolism and of substances surplus to the body's requirement. The liver participates in excretions or in several important ways. Number 1, production of urea. As already stated, that surplus amino acids cannot be stored in the body and are eliminated. The ammonia resulting from this process is extremely poisonous, and it is immediately combined with the carbon dioxide to form urea. This reaction occurs by means of pathways called Arrhenius cycle and is summarized by the following equation. Urea can only be manufactured in the liver. Once formed, it passes into the general circulation and is later removed by the kidneys, finally leaving the body dissolved in the urine. Number 2, breakdown of red blood cells. Red blood cells are continually produced in the bone marrow and have a lifespan of about 120 days. Old red blood cells are destroyed by the groups of phagocytic cells together, known as reticular endothelial system. These cells occur in many different parts of the body and are represented in the liver by the Kupffer cells. All reticular endothelial cells break down hemoglobin obtained from the red blood cell to form a yellow pigment called bilirubin. This is released into the bloodstream and is responsible for the yellow color of the blood plasma. Iron is sent from the storage in the liver, while the protein residue of the hemoglobin molecule is broken down into the amino acids. The hepatocytes cells of the liver extract bilirubin of the blood and transfer it to the bile, forming the major part of the bile pigment. The bile pigments are released into the small intestine along with the rest of the bile, but have no digestive function. Bilirubin is converted by the bacterial action into the stereobilingualism.